The University Medical Center, Hamburg, Eppendorf. Florian.
Thank you very much. I would like to start by thanking the organizers for inviting me to this beautiful meeting. I enjoyed it a lot, and it's also nice to escape rainy Hamburg and spend some days in sunny Sydney. Okay, this is not the first one. We go back to... Okay, there we are. Yeah. So I would like to share our work on a tissue engineering approach for cardiac regeneration with you. I think we've heard beautiful talks that the adult heart has a very limited regenerative capacity. And we've also heard beautiful strategies how to tackle this problem therapeutically. We try to deliver new myocytes to really remuscularize the injured heart. This is not a brand new strategy. It has been already described in the 70s by the Oberbriller group, who transplanted minced newt hearts in this study, and then it has taken a step further by Laurent Field, who transplanted rodent cells in this beautiful study. We have a slightly different approach, though. We transplant engineered heart tissue, which is a three-dimensional tissue construct consisting of a fibrin matrix and cardiomyocytes derived from induced pluripotent stem cells. And in a previous proof-of-concept study, we have transplanted two of those stripe-formed engineered heart tissues in a guinea pig model, and then followed these animals over 4 weeks, and we could see that the transplantation of engineered heart tissue leads to a partial remuscularization of the injured heart, as seen here. So that's the left ventricle scar and the newly formed human myocardium, as shown by a staining for human Q80 in an adjacent section. And this eventually improved left ventricular function. So how to pursue this work? And we're currently working on 3 topics. First topic is cell production. Second one is optimization of patch geometry and development, and third are different transplantation studies. And I would like to talk about this part and show some results on our transplantation work in a small animal model and really preliminary work on a pig study. So, as I said, initially we transplanted these stripe-formed engineered heart tissues, and this was basically because it is our standard model that we use for drug toxicity and disease modeling. However, this model has obvious limitation when it comes to upscaling. So we combined different ideas to come up with a new geometry, and these is engineered heart tissue patches, just that for a small animal model we develop in a 6-well format, and they are 2.5 by 1.5 centimeters in size and consist 15 million cells. And then after about 10 to 15 days in culture, these patches coherently start to beat. In previous upscaling approaches, we always encountered 2 problems. We think that the new geometry has mainly 2 advantages. The first one is already visible here. The pores create force lines, and the cardiomyocytes align along those force lines and mature, and this becomes even more visible in a high magnification, where you can see that the initial immature roundish cardiomyocytes elongate and show regular sarcomeric assembly in the 3 weeks culture period. And the second problem was always cell distribution. And with this new geometry, we see a more homogeneous cell distribution, whereas in the previous approaches, cells mainly aligned along the outer surface. And this can be seen in a 3-dimensional reconstruction. Can we? Going back. Can we? So we see a rather dense and homogeneous cell distribution within these constructs. I've told you that we see synchronous beating after about 10 to 15 days in culture, but initially we see something like this. We see this wobbly beating pattern starting at around day 7, and we always interpret these as competing pacemaker cells, and then eventually one takes over, and we see the synchronous beating pattern after about 15 days. And that's the time point when we start to measure contractility, and we're doing this by using a speckle tracking approach developed by Christine Mummery's group called muscle motion. And we see that over a 5 days time period, contractility remains stable and then increases over the next 5 days, and this is the time point when we use them for transplantation. However, this gives us only arbitrary units, and we have also done organ bath measurements, and we see that under baseline conditions, these patches develop forces between 1.8 and 4 millinewton, and they show a physiological response upon beta-adrenergic stimulation with isoprenaline with a EC50 of about 5 nanomolar, which is radiosensitive. We've done electrophysiological characterizations by sharp microelectrode action potential measurements. On the left-hand side, this is an original recording from one of the EHT patches, and for comparison, this is an action potential recordings from the left ventricular tissue. And they look pretty similar, and this is more obvious when we overlay them. And this was surprising for us. Previously, we have seen more immature action potential shapes in these constructs, and the quantification shows that there is no difference in takeoff potential. That's the potential here. There's no difference in the action potential amplitude. Upstroke velocity is still slower, but there is also no difference in the action potential duration. So how does this more mature phenotype, well, at least more mature than we have previously seen in these tissues, how does it translate into a animal model when we transplant those patches? The animal model that we use is a small animal model, the guinea pig model, where we induce a cryo injury, and then 1 week after induction of the injury, transplant one of the engineered heart tissue patches. This is the transplantation, and then we have a follow-up of 4 weeks. And during these 4 weeks, the engineered heart tissue patches integrate in the epicardial surface. You can still see the sutures here and here. And this is the histology against dystrophin staining, left ventricle, scar, and transplantation of these engineered heart tissue patches leads to large human grafts, as shown by a staining for human Q80 in an adjacent section. And this is consistent. We really see large grafts in these patches, larger than we've seen before. With this, I would like to thank you for your attention. I would like to acknowledge the people in the lab who did the work. The optimization of patch geometry was mainly driven by a postdoc, Marina Reinsch, in the lab. Small animal work is done by a cardiothoracic surgeon, and Lisa Castro. And the large animal work we're doing together with the colleagues in Munich, Christian Kuppert, Andrea Beer, and Markus Krane. Thank you for your attention.
Okay, questions?
Hi, nice presentation. I just got a question about sort of the morphology and maturation of the cells after you implant the patch. So even after some time in vivo, they don't seem to fully mature and look like the host myocardium. Has anyone developed a strain reporter or cell tension reporter or something to see if those myocytes are just not under the same mechanical loading as the heart, and that's the reason? Can you please comment on that?
Well, to my knowledge, nobody has developed such a reporter.
Have you got any inference from like maybe some MRI data or something to see what sort of strains and things they're under?
Yeah, we have developed an MRI protocol, or we're in the process of developing an MRI protocol for this. So far, it's too early to comment on it.
Okay, thanks.
So the question, it's a beautiful study, by the way. So the question of how the graft survive 4 weeks after the transplantation, I think it's not a problem for 4 weeks after the transplant. The real question is how the graft survive the first 4 hours. The 4 weeks after the transplantation, we see the spreading of pre-existing vessel goes into the grafts, have vascular density similar to the naive heart. That's the pig by dynamic culture or the immunodeficient mice. It's a similar observation. The real question is how these the graft, the tissue patch survived in the petri dish for days and weeks with everything they need, and the transplant into the recipient heart, all of a sudden they don't have the circulation, but they do survive. So, but this is consistent for the field need to know that they can survive for the first 4 hours, and then once the first 4 hours, we all know the heart is very robust. Heart has a very robust angiogenesis potential. So,
I agree. This is an important and interesting question, and I don't have an answer. I think that what we see as survival is not just survival, but as I said, the net result of really cells that die, because there clearly is cells dying after transplantation. And I think that a part of the cells survive and then proliferate. And despite the rather mature action potential shape and like elongation of the cells, I think they only survive because they're basically still immature or somewhere in between. But how, and I can't answer how exactly it works.
A nice talk, Florian. Just a quick question, technically. Did you open the pericardium, suture the tissue to the epicardial surface, and then close pericardium? Was that what?
No, in the guinea pig model, we open the pericardium when we induce cryo injury, and then we can't close it in the small animal model. And the pig we do open.
And then close it in?
Well, and then we have a pericardial patch directly onto, and then we close the endogenous pericardium on top of that.
Okay.
Yeah.
Thank you, Florian.